So as Lori said, I'm Brian Berridge. I'm the co-chair of the technical committee, and the technical committee actually encompasses a lot of different efforts, and I'm going to kind of try to give you some insights into some of those efforts today. So before I get into the trees here, I want to talk a little bit about the forest, because I'm sort of a big picture guy, which is why I didn't raise my hand when we were talking about numbers. And I apologize, because I was very interested in your numbers, but I'm a big picture guy. So I want to talk a little bit about context. So the threads, the themes that I hope you'll see threaded through here will be context and translation. And I'm going to try to walk through that, and maybe that'll be obvious as I get through this. So from a cardiovascular standpoint, cardiovascular disease is a leading global cause of death, accounting for over 17 million deaths per year. In the United States in 2011, it accounted for 1 in 3 deaths. Cardiovascular diseases claim more lives than all forms of cancer combined, with cancer being the number 2 cause of death. But it's not all bad news, right? So with a lot of chronic diseases we're seeing, and some significant medical advances, we're starting to see some changes in the trends of the deaths due to those diseases. And in fact, with cardiovascular disease, again, with a lot of these very common diseases, we're seeing a downward trend. So as a medical community, we're getting much better at addressing these issues, and we're actually allowing patients to survive longer. But that has consequences, and that has consequences that are impactful for us. And as a consequence of that, and as you can see from this prevalence data, we increasingly have patients who are living longer with these diseases, right? So that is a contextual influence for those of us who are interested in things like environmental and pharmaceutical safety. And so what you note from that graph is that those of us who are lucky enough to live to 80 or better, most of us will have the privilege of having cardiovascular disease associated with that. So if you look at the risk factors that typically are associated with driving this rather large incidence of cardiovascular disease, there's a number of very common cast of characters in there, like family history, smoking, diet, physical activity, inactivity, diabetes, those kinds of things. But what you don't commonly see on those lists, but we're increasingly aware of, is that there are influences that come from the environment. So, for example, air pollution is a significant cause of cardiovascular risk associated with it, and even pharmaceuticals. And there are pharmaceuticals that have historically been associated with cardiovascular disease, and in some cases, those have been considered manageable risk, and we'll talk a little bit about cancer drugs here in just a little bit. But as our scrutiny of in particular, long-term risk goes up, we recognize that a number of pharmaceuticals have cardiovascular challenges associated with them. Now, changing context a little bit, so those of us who are in the drug development business, one of the things that we angst over every day is our considerable attrition, which is leading to a significantly increasing cost to develop drugs, which results in a significant increase in cost of buying those drugs for patients, which is increasingly making the news and has become a real issue. Well, if you look at, is there a way for me to use the mouse? Just use my finger here. But then I have to be, well, I didn't want to bias by pointing to one or the other, so I'm going to try to do it this way. Then you get people who will feel offended because I pointed to this side and not that side, those kinds of things. But anyway, so if you look across the causes of attrition, safety is represented in these red parts of these pie charts. It is a significant contributor. And the difference between these two columns is actually a trend from 2008 to 2010 data to 2011 to 2012. And the moral of the story here is that trend is increasing. Right? So attrition related to safety concerns is increasing, and in this particular data specifically is dealing with clinical attrition. That's a very, very expensive place for drugs to fall out of development. If you look more closely at what's driving that safety attrition, and this is the oft-cited Laverty paper, and it looks across a number of analyses, and cardiovascular is a pretty significant contributor to that safety-related attrition, and oftentimes, depending on the analysis, actually outpaces hepatotoxicity as a primary cause of safety-related attrition. So the bottom line here is that we have a growing population of patients with cardiovascular risks, and a growing number of pharmaceuticals that have cardiovascular safety concerns associated with them. And so that intersection is actually a real challenge for us. But that intersection is actually the driver for and the impetus for the existence of the Cardiac Safety Technical Committee, which has as its mission the intent to improve public health by reducing unanticipated cardiovascular-related adverse effects from drugs or chemicals, and in particular to develop innovative approaches to support early detection and prediction, and as well as increasing our understanding of cardiovascular toxicology and pathobiology, which allows us to design better drugs for the future. So our approach is very much like any HESI working group, which is where we try to leverage a global multidisciplinary community of good scientists. In this particular case, we've got a lot of cardiovascular scientists who are very interested in that non-clinical to clinical bridge. Uniquely within our group, we're dealing with a subject area that historically, at least within pharmaceutical development, has had some segregation. So the areas where we look very critically at functional assessment and functional cardiovascular risks has, by organization, been disconnected from where we look at structural changes. In our working groups, we've bridged those two, so that's been an important integration part of what we do. Where the rubber meets the road for us is refining our current approaches, so doing what we do better, in addition to developing novel approaches, and these include things like biomarkers, some mechanistic approaches, again, which I'll talk about, and even considering some alternative modeling opportunities. So this is sort of the Cardiac Safety Technical Committee org chart. There's 4 specific and defined working groups. The pro-arrhythmia working group is sort of our legacy organization. It initiated probably about 15 years ago now and was largely built around the QT prolongation torsade risk that was associated with a number of marketed compounds. From there, we developed a focus on biomarkers. So I'm moving this mouse and it doesn't actually show up on these screens, so I don't know why I'm wasting my time. Anyway, the cardiac biomarker working group emerged probably, I don't know, a good 10 years ago. It's probably even been longer than that. Initially had a focus with troponins and did a very good job of establishing a great foundation around troponin as a biomarker to be applied in the non-clinical arena, and is now focusing on biomarkers of procoagulant risk or thrombosis. The Integrative Strategies Working Group on your far right was one that developed 4 or 5 years ago, and again, that was a place where we were trying to bring this structure-function integration together. And so we've got a number of work streams within there. And then more recently and largely driven by technical capabilities, the Cardiac Stem Cell Working Group. And so what I want to do is kind of walk through some specifics of those different working groups and try to give you some sense of how they're sort of working across the issues and in many cases, oftentimes partnering. So as a backdrop for that, even when I saw this slide, and I'm in the middle of all this, I was really, really impressed with the number of participants that we've got involved with these working groups. It's considerable. We have an advisory panel that largely is composed of the leads for those different working groups, plus some additional members like Dr. Frank Selke, who represents sort of the clinical community, Ken, who's been involved with cardiac safety working group things for a very long time. And ultimately, that's all supported by an incredible HESI staff without which most of this wouldn't happen. So getting into some specifics around the different working groups, as I said, the pro-arrhythmia working group is probably it is the legacy organization within the Cardiac Safety Technical Committee. Historically, their focus has been on the translation and in particular understanding the concordance of what we do on the non-clinical, in our non-clinical testing and how that relates to what we see in patients, and I'll talk a little bit more about that in a minute. More recently, they've developed an additional work stream that is intended to sort of redefine the way we look at pro-arrhythmic risk using some mechanistic assays and some novel capabilities around stem cells. So from a translation standpoint, what you see happen in a lot of our working groups, the way that was initiated was to basically take advantage of the work that had already been done. So they did a rather intensive literature search to understand what was published around the concordance between non-clinical and clinical QT measurements in particular. So for those of you who don't work in the cardiovascular field, QT prolongation is sort of the surrogate measure that we use for torsade de pointe risk in the proarrhythmia risk assessment space. And so a lot of what we do in the non-clinical side is actually looking at and measuring QT in our ECGs, in our non-clinical models. And so again, they were looking at the concordance to understand the predictivity of what we do non-clinically for what actually happens in patients. And in fact, there's a publication that has emerged from that. In addition to that, they initiated sort of a unique effort with gathering a lot of data from the FDA and looked at a lot of IND packages, again, to dissect out and look at that, the non-clinical versus the clinical data, and then ultimately compared that to past experiences, and in fact have shown that in fact with the institution of the ICH S7B, which is basically the guidance around the standardization of how we do this in the non-clinicals, they've seen an increase in concordance, sensitivity, specificity for those assays. And so what that does is increase our comfort, our belief in those assays as we do them, that in fact they do reflect what is likely to happen in patients. So again, more recently, having said that, let me say this as a backdrop, having said that, and there is good concordance, there's still an expectation requirement for what's called a thorough QT study in clinical development, and that's generally a standalone study. Where that gets placed in the development cascade is largely driven by the risk that's defined in the non-clinical side, but it's expensive, but it also illustrates the clinical concerns around this particular risk. So as a mechanism for potentially being able to not do that study, they've initiated a novel work stream, and that's using cell-based capabilities to sort of redefine how we look at proarrhythmic risk. So in this particular case, they've got 3 core pillars. One of those pillars is looking very discreetly at the ion channels within cardiomyocytes and understanding the drug interactions with those ion channels. The next part of that is to take that data and to integrate it in silico, and then ultimately test that integration with cardiac stem cell-derived cardiac myocytes, human-derived cardiac stem cells, and to test the proarrhythmic signals that they get in that test system. That has ultimately led to a very natural partnership with the Cardiac Stem Cell Working Group, which has, even though there has a scope, they have some broad interest in how you could use cardiac stem cells in our safety assessment paradigm. Their initial focus is actually in looking at electrical activity and proarrhythmic risk. And again, they've initiated a specific focus in understanding the potential usefulness of those cells, have initiated a multi-site study where everybody's sort of doing a very similar experiment. So to look at the reproducibility of those assays and the sort of fit for purpose of those cells for this particular application. So that will provide a useful foundation for understanding how those cells might be used to complement what we currently do in non-clinical cardiovascular risk assessment. Part of that is looking at a couple of different technologies, microelectrode arrays and the voltage sensing optical. They're looking at slightly different parameters, but ultimately giving you some measure of disturbance or dysfunction in electrical activity and repolarization defects, which ultimately leads to the risk of most concern. So the next working group is the Cardiac Biomarkers Working Group. And again, this is one that has evolved from an initial focus in troponin to one that is currently focused on procoagulant or thrombotic risk. So one of the things that we struggle with in the cardiovascular safety arena is that some of these clinical cardiovascular issues aren't discrete toxicities like we often describe non-clinically. So for example, You take a compound into large clinical trials or even post-marketing, and you're starting to use that medication in a lot of patients with a very varied background. And you go back and you look at the incidence of what they term major adverse cardiovascular events in those studies, and you find out that there's an imbalance. So people who are on those drugs have more events than those who don't. Well, many times those events are things like heart attacks, strokes, transient ischemic events, those kinds of things. Well, one part of those syndromes is actually coagulation and thrombosis of either coronary arteries or potentially vessels in the brain. Well, as a routine, we look at coagulation parameters in our non-clinical studies, in our animal models, platelet counts, coagulation times, those kinds of things. But we don't look very sensitively for potential issues that might represent a procoagulant risk, right? It's either you've got thrombosis and some change in your coagulation or you don't, right? It doesn't catch the sort of less discrete events that in a susceptible patient might play out badly. So that's in fact what this group is doing. And so what they've done is initiated a couple of proof of concept studies, one of which has completed. And so in this particular case, they did a rat study. They used bacterial endotoxin as sort of their toxin, which is known to cause procoagulopathy. They looked at a variety of biomarkers, and they were able to look across those biomarkers and understood those that changed and those that didn't. They've put together a manuscript that I'm soon will be published. So now what they're doing is going that next step, right? So that's in our traditional model with a rather severe challenge to it. So now they're going to go into an animal model that represents a more traditional patient risk, right? So go into ZDF rats. So these are animals that are obese, they have metabolic syndrome, they're diabetic. They're going to use doxorubicin as their challenge. That's a drug that's still widely used. And so they're going to look at a more translationally relevant model and use some of the biomarker data that they generated in the first study to again, look for evidence of thrombotic risk in that model. So now transitioning over to the Integrated Strategies Working Group, and this is actually the one that I'm most involved with. A number of years ago, and this had come from the fact that within the biomarker working group, we had people who were traditionally dealing with sort of structural assessments, lots of clinical pathologists and some anatomic pathologists like me involved in that group. And then in the proarrhythmic working group, we had folks who were safety pharmacologists typically associated with functional assessments. Well, as you start to get outside of your box, as it were, and start to talk, we recognize that there really was a need to integrate across those traditional lines. And that's ultimately where the Integrative Strategies Working Group developed. So as we looked at our respective areas, and in particular the intersection of those areas, there were two work streams that emerged. One was an interest in measures of contractility in our non-clinical model. So best established in safety pharmacology, again, is the proarrhythmic risk evaluations. And in fact, many of you are probably aware that part of a GLP safety package is a non-rodent cardiovascular study that includes very sensitive measures of things like blood pressure, ECG. Heart rate would be included in there. What's not as consistent are measures of contractility. But what's interesting about that is that when you talk to clinical folks who are going to ultimately translate the things that we describe in our non-clinical studies into a clinical trial, the thing, regardless of how you describe your issue, right? Changes in troponin, I've got myocardial necrosis or some degenerative process or inflammation, whatever that is, they immediately start thinking about function because that's what clinicians do, right? They measure function. They don't go in there and cut out pieces of the heart and look at it histologically like I do. They think about function. And contractility is the primary function that they're thinking about. Well, we don't traditionally, haven't consistently put measures around contractility. So as we recognize this disconnect, and there's been more focus. Now, it's not that we don't have the ability to, and in fact, some people do, but there never really had been a characterization about how you do that consistently, What's the reproducibility among sites? What's the sensitivity of being able to do that? What's the predictivity for changes, either in increases or decreases in contractility? And as a result of that, when you submit a package and you've got that data in there, the people on the receiving end of that, oftentimes are the regulatory folks, don't know what to do with it. If they don't know what to do with it, they just ignore it. So you're just generating data without any real utility to it. And it's data that has an important part to play in the clinical translation. So that particular group set out to do something about that. On the other side of that was recognizing that our traditional animal models don't look a lot like patients. And so we start to explore the idea of maybe there are other ways to model risks non-clinically that might have some more relevance to patients. So specifically around the contractility, and apologies for the small size of that print, essentially they set out to do a cross-site study with a select group of positive and negative inotropes, basically pharmaceuticals that change contractility. And these were drugs that had known clinical effects, and so they could look at that concordance. And they did it across sites so that they could look at the reproducibility. And ultimately what they found was that in fact, well, this is the basic description of the study that they intended to do. Again, they used positive and negative inotropes. They did a cross-site study, and then ultimately they put the statistics to the data. And what they found, not unexpectedly, is in fact in a telemetered conscious dog model, which is typically what is done for a GLP cardiovascular safety practice, that you can generate these measures. They do have concordance what happens with patients, and they have reproducibility. In fact, the data was very, very close among those sites. So now what this group is doing is going to the next logical step, which is actually to put those kinds of measures in the context of the repeat dose studies that we do in our general talks, right? So this is, again, bringing that integration of structure and function. Those repeat dose studies are where we traditionally do those structural assessments. Those repeat dose studies are oftentimes where a lot of our toxicities emerge with that repeat dose paradigm. Those repeat dose studies are where you typically see those problems in patients. So now we're going to take that very important measure, put it in the context of where we typically look at the structure, and we'll have a much better integration than we do with traditional practices. So transitioning from there, the conversation around alternative animal modeling. So again, what we recognize is that patients are very, very complex, and traditional animal models of safety are not so much, right? But that's to some degree by design. What we're interested in are fairly discrete measures of whether there's toxicity or not, whether there's adversity or not, and what a dose level you're seeing those changes, right? So we don't want to have a messy model, or you're going to have to use hundreds of animals to be able to dissect through those issues. So there's a good reason why we do what we do, but as a consequence of that, they're not very reflective of the human condition. The other thing is that, as I described earlier, chronic toxicity in aging patients is where our growing concerns are. Well, we know from the Olson concordance paper of 2000-2001 that our cardiovascular assessments are really, really good, but mostly for the acute stuff. So there really is a need to sort of explore how we can model those chronic cardiovascular risks other than treating lots of patients for years and then stepping back to see what happened. And that's the kind of thing that we've been exploring in the alternative animal modeling space. But as you can imagine, that's not an easy conversation. The fact of the matter is that there are animal models of cardiovascular stress and disease that represent, some similarities to patient comorbidities. There are changing attitudes in the safety assessment community about acceptability and tolerance of looking at alternative models. But it's not easy to define which model and for what, right? So just a few papers to represent those issues. Again, the increasing in acceptability of entertaining the idea of using alternative models. There is precedent for using disease and stress models, mostly in academic or very experimental settings. But in fact, we realized that if you use an altered or cardiovascular dysfunction model, sometimes you see things happen that don't happen in a normal animal. things may be relevant. So those are the kinds of things we'd like to understand better. So what we've done is a couple of things. One, as we had these very challenging discussions, and they always ended up in the same place with, yeah, sure, you could do a dobutamine stress, you could do a transaortic constriction and get a hypertrophied heart, and that would certainly be an altered state. You can use spontaneously hypertensive rats. You could use ZDF rats with metabolic syndrome, all those things, but which one is relevant? You know, so are we just going to ultimately translate our lack of concordance from one space to another? So it's very difficult to know which direction to go in there. So in the context of those discussions, one of the things that we realized was that, well, in fact, we know a fair bit about comparative cardiovascular physiology that might actually influence how we look at the data that we get from those different animal models. But when you went to the literature, there wasn't an easy place to go and find a single source that would explain differences in heart rate, blood pressure among species, differences in calcium utilization, ECGs, and all those kinds of things. Even though everybody knows you don't go looking for QT prolongation in a rodent, that a rat heart rate is a whole lot higher than your heart rate, and if it's not, then you please need to leave this room and go seek medical attention. But there are very, very distinct differences. But those differences aren't often considered in the context of the data that we generate, right? But how many of us have dealt with projects that were shut down because you had a rather severe myocardial change in a rodent for which you didn't have a dose margin, but you didn't know if it was a rat thing or not? So again, what we're intending to do is to sort of pull all those pieces together and try to provide that as a sort of a contextual background for how we look at the data that we get from the animal species that we got. The other thing that we're doing is trying to define a space that we can go in and actually start to explore some of these alternative models in a space where there might actually be less concern about generating data that we won't know what to do with, or a place where historically we've had a little bit more tolerance for cardiovascular risk. And in fact, that space is in the cardio-oncology space. So for those who aren't familiar, so in cancer treatments, another space where we've gotten a whole lot better, I'm going to talk about that here in just a minute, cardiovascular safety is a growing concern. And historically, it's been a recognized issue, and we've accepted it because of the severity of the condition. So this is a place where we've got a fairly well-defined patient population. We've got tolerance for risk that doesn't exist in other therapeutic areas, and potentially as a space, and also a clinical community and a patient community that is increasingly interested in having opportunities for having safer medications for the future. So I use this slide to illustrate the point, and actually it's two different sets of data, major tumor mortality trends between males and females, and the important part there is that, again, we're getting better at treating those cancers, and so patients are surviving longer. And again, the consequence of that is that you have patients living longer to experience the consequences of the drugs that were used to treat the cancer. Okay, so again, as that happens, so this is another example of where no good deed goes unpunished, right? So, all right, that's fantastic. You've saved me, but now I got heart failure, so I'd just soon not have that too. So can you please develop something that's a little bit more safe? And so that's the kind of thing that we're interested in. Again, it's been recognized that cardiovascular risks exist within cancer drugs. Doxorubicin is a perfect example, classic drug-induced cardiotoxicity. To this day, with all the experimentation, still not a clearly defined mechanism associated with it, but it's there, but we accepted it because of what it did for cancer. A new, relatively new class of cancer drugs, also very, very effective, driving much of this trend in increase in patient survival, or the tyrosine kinase inhibitors, and we're increasingly see evidence where some of those drugs are associated with cardiotoxicity as well. So this is actually a nice image that Cyril put together, and I think it illustrates some interesting points, right? So the good thing is that we're saving patients. The bad thing is it might be at the expense of their heart. The fear, and we've had some conversations with oncology and cardiology folks, and in fact, that communities are coming together really, really nicely on the clinical side. The fear is that nobody wants to give up progress in the treatment of tumors to get over conservative around cardiovascular risks, right? And that's perfectly understandable. But that is a space where the expectations are changing, and so we need to change with them, and that's where the opportunity lies. So with all that, right, so we've got increase in populations of patients with cardiovascular disease and risks and sensitivities associated with them. We've got increased expectations around safety. In the background, there's this other issue that's developing, and that is a growing critique of the use of animals for biomedical research in general and safety assessment in particular. And so that's sort of a growing thing, and one of the things that we all recognize is that in the world that we live in, animals are an important component of the assessments that we do, and we use them because we believe that they have relevance, and in fact they have done added considerably to our protection of patients over time. But this is not to be ignored. And in fact, those of you in Europe are probably very much aware of this. This sort of came to a head a few weeks ago, where the European Commission, because of a process they have in place where citizens can gather signatures and ultimately have an issue put in front of the Commission, and they have to deal with it because that's their agreement. There's actually a group that brought forward, they had over 1 million signatures, I believe, and they put together a proposal to completely stop vivisection, right? So it's getting to that point. So the risks to our way of doing our business is real, and it's growing rather quickly, so it's not to be ignored. Well, I think that provides another opportunity, right? So that opportunity actually is for some increase in integration within HESI. So there's a relatively novel subcommittee or working group that developed last year out of the emerging issues, and it's all about how you develop non-animal alternative methods for safety assessment. So this seems like a really, really good time for us to start partnering with that group to look critically at how we do our business before somebody takes away our ability to do it. So in summary, the outputs of the group include things like manuscripts, and there has been good steady progress and a steady flow of manuscripts coming out of these working groups. We also do a lot of outreach and education associated with different presentations at different meetings that are listed there. So in summary, the HESI cardiovascular safety efforts are world leading, and they're unique in their sort of multidisciplinary, very integrated approach. You can find cardiac safety efforts in other organizations, but you won't find them like you find them within HESI. We've got strong engagement of a very multidisciplinary group that includes the tripartite that HESI is committed to. We've got very effective communication strategies and outreach. We give lots of presentations to lots of groups. I think we're best positioned to advance and improve on our current approaches, and I think we're also best positioned to define a different future. And I think that a different future is something that's inevitable. I think it ultimately could lead to an improvement in the way we do our business, but it's certainly going to stay ahead of the risks associated with concerns of doing it the way we do it today. And with that, I'll conclude.
Two questions answered at the same time. One is, could you have just done this kind of work? Could any company do this work within its own walls? And the second part of the question is there anywhere else other than HESI that you could have done all this?
So the answer to the first question is yes, you could have, but not near as effectively in two places. One is the amount of resource and time to get to the same conclusion, and the other is the level of influence, right? So no one organization could have done this to have the impact that the collective has had, right? And that's part of the game. The second one is I don't know of another organization that has this level of multidisciplinary integration and coordination like we've got in HESI, particularly in the cardiovascular space.
So Brian, I have a quick question. When you're talking about biomarkers and novel biomarkers, you have, for example, the relationship between doxorubicin treatment and cancer and cardiovascular events. Are the efforts to to identify potentially early indicators that somebody should stay away from doxo and use a different type of chemotherapeutic agent because a person has a genetic liability, as opposed to biomarkers that tell you, yes, there's a cardiovascular event associated with initial treatment in XYZ patient?
Yeah, so I think it's two things. One is, I don't see anybody not giving a cancer patient a drug that they think is going to effectively treat that cancer because they think there might be an enhanced coagulopathy or a procoagulant risk. What I do see is the ability to recognize that a patient has that susceptibility and to manage it more effectively prospectively. The other part of that is that I don't think that we have the ability, I don't think we have enough ability on the non-clinical side to help select drugs away from those risks because I don't think we're modeling them very effectively. So I think that in non-clinical models, if we can recognize a susceptibility to thrombotic risk because we see some change in some endothelial activation marker, for example. So not overt thrombosis, but if we know that's happening, and we know that in patients that can translate into myocardial infarctions or stroke or whatever, then I think we have the ability to screen drugs to find a drug that potentially has less of that risk. Does that make sense? Mm-hmm. So I think it's both of those things. Proactively managing patients and an ability to identify safer drugs.
And one final point, when you were talking about biomarkers, I think you had on your listing something that you call extracellular vesicles.
Yeah.
What was that referring to?
So one of the things that they've looked at are these surface blebs from endothelial cells that ultimately circulate. And in fact, vascular injury working groups have looked at them as well. And so they're, I don't know what they tag them with or if they use.
Also similar to like the shedding of
yes.
something in the liver
Yes.
when there's an early event taking place in the liver?
Yes.
Okay.
Yep.
I was just going to comment on Jose's question. Obviously, the paper we published this year, Laura Coe-Smith was the lead author. So that was an AstraZeneca collaboration with the big cancer hospital in the northwest of England, Christmas. And on that point, Jose, the cardiac issue is often very latent, so it doesn't emerge for 2 or 3 years as an issue of the ejection fraction. But what we managed to do in that paper was recapitulate the issue in rodents that we saw in the clinic, and that's now a perfect model to think about screening it out early. And the clinicians on that project were talking about using it. Obviously, you wouldn't deselect patients from the study because they've got no other therapeutic option, but perhaps to manage the regime, to limit dose in particularly susceptible patients. But that work has unfortunately come to an end at the moment, but I really would love to think about how we can capitalise on what we know.
That brings up another really interesting point. So most of the time, cancer treatment are protocols, right? They're not single drug kinds of things. And we don't actually explore the relationship of those things. Like, for example, radiation and how that makes you more susceptible to doxorubicin toxicity or radiation plus doxorubicin plus a TKI. And so if we can understand how to model the risks, then I think we can explore those protocols. Right now, that gets empirically done in patients, which is a little unsettling to consider. But again, it's a space where we've tolerated that because of the severity of the disease. As we get better, that tolerance is going to go down.
And it's that potential for, Andrew Hughes is one of the senior clinical oncologists today, they've made the comment that this would be helped to manage sequential treatment. So if you could model in rodents the things that cause a latent susceptibility